Or do you just say you do? Well, you know. All right, let's go ahead and jump right in here. Okay, so y'all might be surprised by this, but we're behind. I honestly haven't looked at the schedule recently, but I know we're at least one day, maybe more behind, which honestly I'm not really worried about that, okay? But I do need to, so today I'll be reevaluating the schedule and just kind of seeing where we're at in terms of, bless you, in terms of like the exam. I'm 99% sure the exam will be after spring break. I think that I originally scheduled it next week before spring break, but it's going to be after. Because today we'll get to the TCA cycle. On Thursday, we should be able to finish it and maybe start oxidative phosphorylation. But then next week will be dedicated to oxidative phosphorylation. So then you'll have spring break to work through the last problem set. We'll probably have the exam on the Thursday after spring break, okay? And it'll cover everything from intro to metabolism through oxidative phosphorylation. Basically, so it boils down to, this is how we make most of our energy, okay? That's gonna be what the first exam's on. Okay, any questions about that? Yeah. Problem set 4, yes, because that's over the mechanisms of glycolysis, and we've covered all that stuff. Yep. Okay, so the first bit of this today is just reviewing what we talked about on. Yep.
I have a question. So the next problem set
will be due after spring break, yeah. So it's going to be TCA cycle. So regulation is part of this problem set. But then, so the next one will be TCA cycle and oxidative phosphorylation. So you'll have spring break to do it. And I assume everybody will be working diligently over spring break on a problem set for biochem too, right? Gabby's like, oh yeah. Okay. All right. So we talked about gluconeogenesis. We made it through gluconeogenesis last week, and we talked about how gluconeogenesis is the reverse of glycolysis, right? But when we reverse pathways, it's not always a zero-sum game. So glycolysis, we know, nets 2 ATPs and 2 equivalents of NADH. When we reverse this, because we have to reverse irreversible steps, we end up investing more ATP than we produce for glycolysis. So gluconeogenesis consumes more energy than glycolysis produces. Okay, that's one important thing to remember about these opposing pathways. And that being one reason, and another being, like, we simply don't want opposing things happening at the same time. Where we have to have a way of controlling the flux through these two opposing pathways. And this idea of controlling flux is something that's going to be pervasive this entire semester. Every time we think about pathways, we need to think about how we control movement through those pathways. Okay, and since glycolysis and gluconeogenesis happen in the same cellular compartment, they both happen in the cytoplasm, it's really important to be able to turn one off while the other one's turned on, and vice versa. Okay, so we talked about intracellularly how there are signals that tell us about the energy charge of the cell, right? So we talked about how ATP and AMP are the primary signals of the cellular energy charge. And we know that when ATP levels are really high, we want to inhibit glycolysis because we have plenty of energy. But that same signal, we want to perhaps activate gluconeogenesis, or at least not inhibit it, okay? And the opposite's true when we have high levels of AMP. That signals low levels of ATP, so we want to activate pathways that produce energy, okay? And this image here shows us that in the presence of high ATP, we're going to inhibit phosphofructokinase. We're also going to inhibit pyruvate kinase, right? Two of the key steps in glycolysis. So high levels of ATP signaling high energy charge of the cell inhibits glycolysis, okay? But conversely, high levels of AMP, which signals low energy charge, activates phosphofructokinase, activates glycolysis. And the opposite is true for gluconeogenesis. High levels of AMP will inhibit FBPase, the reverse of PFK. So we inhibit gluconeogenesis. So the intracellular signals make a lot of sense. We then introduced this other idea, this molecule F26BP, fructose 2,6-bisphosphate. This is an isomer of fructose 1,6-bisphosphate. And we're going to see, and we talked about last week, how it is made from fructose 6-phosphate in the same way as fructose 1,6-bisphosphate is. It's just a different enzyme that does it. Okay? So what we notice here is that this molecule strongly activates glycolysis by strongly activating PFK, but it strongly inhibits gluconeogenesis by strongly inhibiting FBPase. Okay? And this molecule is the link to the rest of the organism. Okay? It is synthesized or degraded in response to hormone signals. Okay? So there also is a link between the ability of hormones to activate and inhibit enzymes and the overall cellular energy charge, right? So I stressed last week how really small concentrations of this molecule are quite activating or quite inhibitory. So we're looking at the activity of phosphofructokinase here, right? So PFK activity. We see that the red line is with no activator around. So that's the basal activity. We see a Km in like the 2 millimolar-ish range. But adding really small amounts of this activating molecule aggressively decreases the Km. So really small amounts make this enzyme much more active. But that can be reversed by increasing the intracellular concentration of ATP, right? So this molecule, F26BP, will still be activating, but not nearly to the same degree if we have a high cellular energy charge, right? And we won't get into any more of that, but what I really want to stress with this is making sure you understand that there is a linkage between intracellular conditions and what the organism needs, right? A certain cell can't respond to organismal needs if it doesn't already have satisfactory conditions inside the cell, right? If a cell's not healthy, it can't contribute to making the rest of the organism healthy. All right, so moving on. This brought us to the idea that we left off with, and that is how we make this molecule F26BP, okay? So we talked about how it all starts with the blood sugar level. When there's high levels of sugar in the blood, this is recognized by the pancreas and we secrete insulin. And the thing that I really want to stress about insulin secretion is that inside of a cell, this extracellular signal results in the production of protein phosphatase 1. Okay? So insulin activates this phosphatase. And inside the liver, that phosphatase has an impact on the flux through glycolysis and the flux through gluconeogenesis through the activation of this molecule F26BP. Okay? Conversely, low blood glucose levels are sensed by the pancreas, which triggers the secretion of glucagon, a hormone that signals exactly the opposite of insulin. Okay? The important thing to remember about glucagon secretion is it activates a kinase. Okay? So these two hormones are secreted in response to opposing organismal conditions. And they elicit an opposing response inside the cell. Glucagon, secreted in response to low blood sugar, activates a kinase, whereas insulin, secreted in response to high blood sugar, activates a phosphatase. Two enzymes with opposing activities. That's relevant. Okay? If we think about the liver cell, and I really want to focus on the liver because the liver is the organism that ultimately is responsible, not the organ, the organ that is ultimately responsible. She gave me a weird look. The liver is the organ that's ultimately responsible for maintaining the health of all other tissue, right? Especially those that aren't good at making their own energy, like the brain and the heart. Okay? Things that we absolutely are dependent on for survival. So in the presence of low blood sugar, we don't have a steady supply of energy for the brain, and that's a big problem. So the way mammals fix this problem is in the liver, we mobilize other molecules to be converted to glucose. We mobilize glucose. We make glucose from basically anything we possibly can. And then we take that glucose, we secrete it into the blood, and send that glucose to the brain, to the muscle tissue, to the cardiac tissue, whatever needs it. Okay? So it makes sense when there's low blood sugar levels that we activate gluconeogenesis in the liver, because under these conditions, we need to make glucose to pump into the blood to fuel other cells. So when protein kinase A gets activated, this is going to result in the activation of gluconeogenesis. Okay? The opposite's true when insulin's secreted. This is going to result in the inactivation of gluconeogenesis in the liver. Okay? Because if there's high blood sugar, there's a steady supply of sugar, steady supplies of fuel for the brain, for the muscle tissue, for the cardiac tissue. So there's no need to mobilize more glucose in the liver. So we inactivate gluconeogenesis. These two hormones have opposing impacts on flux through these pathways. The way that they work is by ultimately impacting the production of this molecule, fructose 2,6-bisphosphate. Remember, fructose 2,6-bisphosphate activates glycolysis and inhibits gluconeogenesis. Okay? It is made by this dimeric enzyme with two active sites. One of the active sites is a kinase domain. The other active site is a phosphatase domain. The kinase uses F6P as a substrate and it makes F26BP. This results in activation of glycolysis, inhibition of gluconeogenesis. F26BP activates PFK, inhibits FBPase. So when the kinase domain is active, we have active glycolysis, inhibited gluconeogenesis. And as it turns out, this is activated by insulin. This makes sense. When insulin is present in the blood, we know that it means we have plenty of blood sugar. There's no need to mobilize more glucose. So let's inhibit gluconeogenesis and let's activate pathways that can use the glucose. Okay? So this makes sense. The FBPase domain is inhibited by insulin. So the idea here is when let's just move on because this makes it more clear. Okay, so we have our central core enzyme with two domains. We have our PFK2 domain and we have our FBPase2 domain, right? Only one is active at any given time. And the activity of the two domains is dependent on the phosphorylation state. When this enzyme is not phosphorylated, PFK2 is active. The kinase domain is active, and we are actively making F26BP, actively inhibiting gluconeogenesis, actively activating glycolysis. When the core enzyme is phosphorylated, the kinase domain is inhibited, the FBPase domain is activated, and we are breaking down F26BP. So we don't have inactivation of gluconeogenesis, we don't have activation of glycolysis. Okay, that's the really big picture. Insulin activates a phosphatase, which removes the phosphate from our core dimeric enzyme, activating the kinase domain, which leads to the production of F26BP, which leads to the inactivation of gluconeogenesis and the activation of glycolysis. The opposite's true when we have low blood sugar. Glucagon is released, which signals the activation of protein kinase A through our secondary messenger camp. This kinase activity leads to the phosphorylation of our dimeric enzyme, which inhibits the kinase domain and activates the phosphatase domain. This leads to the breakdown of F26BP, which leads to the activation of gluconeogenesis because our FBPase can be active, and we don't have activated glycolysis.
So the job of that's possible.
Which of these activities is active? And it's inverse, right? So when glucagon is active, the protein kinase A is active, which activates the phosphatase domain of our dimeric enzyme. So to me, that's the trickiest part, right? Because I stress that when glucagon is secreted, kinases are active. But that leads to the inactivation of the kinase domain and activation of the phosphatase domain. That's the tricky part, I think. Yeah.
So we're just assuming the phosphate's already there.
Well, it'll be present in one of these two states, right? So when this protein's synthesized, it's synthesized without the phosphate there. It's synthesized without the post-translational modification. So we need to have protein kinase A active to put the phosphate on there in the first place. So the default state would be an active PFK2 domain. Make sense?
Can you repeat what you said about the kinase?
Okay, so whenever glucagon is secreted, this means that we have our promiscuous kinase active, right? Protein kinase A is a very promiscuous kinase. It phosphorylates lots of things. So glucagon activity means kinase activity in the cell. Insulin secretion means phosphatase activity in the cell. What I think is a little bit weird in reverse, and you have to like kind of reverse in your head, is that when the kinase is active in response to glucagon, that activates the phosphatase domain of our core enzyme, right? Because that phosphorylates this dimeric enzyme, which activates the phosphatase domain. So glucagon activates kinases, which activates the phosphatase. That's the tricky part, I think. But if
Activates the phosphatase by adding phosphate.
by adding a phosphate, right? Because that's the job. The kinases, the only thing they do is they use they take a phosphate from ATP and put it onto something else. So in this case, that something else is this dimeric enzyme. Phosphorylating it leads to the inactivation of the kinase domain and the activation of the phosphatase domain, which leads to the degradation of the activating molecule F26BP. So there's a lot to this, right? There's a lot of pieces. But if I truly believe if you think about the big picture, think about what has to happen inside the cell in response to high blood sugar and low blood sugar, it really makes sense. So the phosphorylation site of this dimeric enzyme is not in the active site, right? So like it'd be somewhere else. It's think about it as allosteric activation. It's the same thing as an allosteric site. By putting the phosphate there, it impacts the activity of the two active sites through allosteric communication. So it just basically it kind of like forces an amino acid into the binding site for the FBPase or for the PFK domain. It just impacts how things can function. Yeah, just think about it's allosteric. Yeah. Yep. Any other questions? This is great. Y'all asking questions. I like it.
Look, I just need to stare at it.
It really does make sense if you look at it for long enough. The connections are there. And I was telling Kyla like last week that I've taught this for basically 10 years. This class for like 10 years, and it's taken me 10 years to get to a point where I see the connections well enough that I really think that they really make sense in my head. So hopefully I'm explaining them in a way that it makes sense to me, and I hope it makes sense to you. All right, like nobody ever explained like the connections to me. It was basically like just go memorize this, but the connections are there. The logic is there. You just got to see it. Make sense? All right, let's move on. So the link between different activities in different tissues is important, right? Because when we think about the role of the liver versus the role of basically everything else, the liver does very, very different things, which is why I'm going to stress several times this semester that the gene expression profile in the liver is so very different than other tissues. So we're going to see one example of that today. Because the liver just has a different role. Its role is to make sure that other organs can function properly. Okay, here's one example of this. It's called the Cori cycle. It's the way that the liver and skeletal or cardiac muscle tissue can communicate and support each other. Or really the liver supports the muscle tissue. So the idea here, right, is that when your muscles are actively functioning, let's say during a sprint, right, you need fast, steady supply of energy to your muscle tissue, right? We talked about before how the muscle tissue uses up its ATP supply pretty quickly, and then it can rely on the presence of the phosphocreatine as a supply of energy. But then it starts to rely on anaerobic respiration and then aerobic respiration, right? So for anaerobic respiration to be able to provide fuel, the idea is there has to be a steady supply of glucose. So that glucose comes from the liver. So the idea here is that in the liver, under these conditions, gluconeogenesis is active. we are actively converting pyruvate or other cellular metabolites back to glucose, which gets pumped out into the blood, transported to the muscle tissue that needs it. It can be brought into the muscle tissue, which can then go through glycolysis, making 2 ATPs, producing pyruvate, which then under anaerobic respiration can then be converted back to lactate. Lactate is then pumped out of the muscle, taken back to the liver, where it can be converted to pyruvate. Pyruvate can then be converted back to glucose, and it becomes a cycle. Okay, and this is why when you hear somebody say that your sore muscles are because of lactic acid buildup in your muscles, it's complete trash. It's bullshit. That's not why you have sore muscles. Okay, because you do not acidify your muscles. Lactic acid, number one, you don't have lactic acid in your muscles. You have lactate, and that lactate leaves immediately. It doesn't hang out. You have a question? Yeah, yeah, exactly. So it's the process of repairing your muscles. All right. So and what'd you say? Oh, I mean, like, but that's what's so like textbooks say that, but it's wrong, right? Like conventional logic is the buildup of lactic acid is what causes what causes muscle fatigue, but it's not. It's not what gives you sore muscles because the lactic acid leaves immediately so that you can power the cell in other ways.
So like a completely hypothetical. So like say the lactic acid did stay in the muscles,
Mm-hmm.
you feel even more like discomfort.
So I don't know the answer to that because it doesn't happen.
I know, but like hypothetically.
I mean, maybe because I mean you can acidify the blood, but the blood is buffered. The cells are buffered, right? So to say that you can have conditions where the lactate becomes lactic acid and builds up is just not true because it's buffered. So I don't think so, but I don't know the answer for sure. I mean, because yeah, so when you start acidifying your blood, there becomes, I mean, it's not muscle fatigue, it's death.
Yeah.
So we want to avoid that. All right, so here's kind of another example, and this one explicitly says like this is during a sprint, right? And what I want you to notice here is that this one's a little more complicated, and it talks about the linkage in other pathways. Okay, so here we're not just thinking about the Cori cycle, we're not just thinking about anaerobic respiration, we're thinking about other things. So in the liver during a sprint, we know that we want to mobilize glucose. So we have gluconeogenesis active, represented by these thick arrows. Okay, glycerol. Glycerol, we can make this by breaking down some amino acids. We can make it by breaking down fats. Okay, so glycerol can be pumped into gluconeogenesis to make energy. Amino acids can break down into pyruvate or into other glycolytic intermediates, so they can fuel gluconeogenesis. Okay, everything in the liver is diverted to make glucose. Glucose is pumped into the blood, and then it either goes to the muscle tissue or cardiac muscle tissue, right? It makes sense for cardiac because the heart's pumping really, really hard to move the blood, so you can move oxygen and move glucose to other tissue. Okay, in cardiac tissue and in the muscle tissue, we have glycolysis active. So glucose is being actively broken down to pyruvate in both tissues, and muscle tissue and liver hepatocytes are really the only cells that store glycogen, right? The storage form of glucose. So in all of these cells, we have glycogen breakdown being active because in all of these cells, we want to rapidly produce glucose. So glycogen breakdown is activated. Okay, in the muscle tissue and in the cardiac tissue, we are doing everything we can to make energy. In muscle tissue, that involves primarily anaerobic respiration because we don't have a really steady supply of oxygen. Okay, but we do have some aerobic respiration that produces a lot more energy. What's interesting here is in the cardiac muscle cell, you see no indication of anaerobic respiration, right? And hopefully that makes sense because you have a massive influx of oxygen at any time, right? From the lung, I don't know if y'all take on the anatomy or physiology, but right? When you breathe in, the first stop for that oxygenated blood is your heart, which can then be pumped elsewhere, right? So the heart has massive supply of oxygen, so there's no need for anaerobic respiration because it's actively respiring. So it's cool because it shows how these different tissues all have integrated metabolism. The liver being polar opposite for the most part of all of the tissues, because its role is to make sure these tissues are able to function properly. All right, let's spend a little bit of time talking about a couple other sugars, right? So two other sugars that commonly come from our diet, fructose and galactose, right? Galactose, anybody know where we get galactose from? Milk, yeah. So like, lactose is a glucose and a galactose disaccharide. Where do we get fructose from? Fruit, high fructose corn syrup, and like sucrose, table sugar. That's fructose and glucose. Okay, so the high fructose corn syrup is a huge one, right? And what we're going to talk about here kind of shows you, at least metabolically, why why having sugar in the form of high fructose corn syrup actually matters. Okay, so in most tissues, matters bad. Be patient, Sam. In most tissues, fructose is broken down, is catabolized in a very similar, essentially identical way as glycolysis, okay? Because hexokinase is loose enough in its specificity that it can use fructose as a substrate just like glucose, right? Hexokinase is the first enzyme of glycolysis. It will also phosphorylate fructose to make fructose 6-phosphate. So in most tissues, this is the path for fructose metabolism. Fructose gets phosphorylated by hexokinase to make fructose 6-phosphate, an intermediate glycolysis. So then this proceeds through glycolysis like anything else. Okay? Overall, the energy production from fructose is identical to glucose. We use 1 ATP to make fructose 6-phosphate, just like we use 1 ATP to make glucose 6-phosphate. Energy yield identical for fructose and glucose. So that's good. They're both 6-carbon sugars. They both have the same overall oxidation state of carbons, average oxidation state, so it makes sense they produce the same amount of energy. However, things aren't quite so simple in the liver, right? And remember, the liver matters. The liver really, really matters. For one thing, it's the first stop when you eat something, right? So you eat a heavy fructose diet. The fructose goes into your blood, the first stop is the liver, and the liver decides what to do, okay? So here's the problem with that, is that in the liver, the gene expression profile is different. The liver does not express hexokinase. It expresses this enzyme glucokinase, which is very specific for glucose. So in the liver, there's not hexokinase, there's glucokinase. And that simple issue diverts fructose metabolism down a different pathway, okay? So instead of fructose being phosphorylated to fructose 6-phosphate, it uses the enzyme fructokinase, which phosphorylates to fructose 1-phosphate. This is not a glycolytic intermediate. That matters because the next step in fructose metabolism in the liver is using aldolase. So we don't make a fructose bisphosphate. Instead, we immediately go to the aldolase, which converts our fructose 1-phosphate into dihydroxyacetone phosphate, a glycolytic intermediate, which can be converted to GAP, which proceeds through glycolysis, and it converts it into glyceraldehyde. Note this is not glyceraldehyde 3-phosphate because the enzyme used fructose 1-phosphate, not fructose bisphosphate as a substrate. So we end up with our aldehyde not having a phosphate on it, which isn't a problem. There's an enzyme called triose kinase which can actively phosphorylate it to make GAP. Okay, GAP can then proceed through glycolysis. So big story here, right, is that 1 fructose in the liver proceeds through glycolysis and has a net energy yield the same as glucose. Okay, we still invest 2 ATPs to get to our payoff phase. We don't lose energy. Okay, but the really, really, really big problem here is that proceeding down this path bypasses PFK completely. Remember, PFK is phosphofructokinase. It is the key regulatory point of all glycolysis. So by proceeding down this alternate pathway, we proceed through a metabolic pathway that does not have any kind of control. There are no control points in this pathway, meaning it doesn't matter if your entire body has plenty of energy, it's going to make more ATP for you. But it doesn't make more ATP. Instead, it results in the buildup of acetyl-CoA in the cell, and the buildup of acetyl-CoA tells your body, tells your liver to make fat. Okay? a buildup of acetyl-CoA results from fructose metabolism in an uncontrolled way. That produces acetyl-CoA buildup, which results in fat synthesis. So it really does matter, right? When we eat sugars that contain fructose, like sucrose, there's not a mass influx of fructose into the blood because the slow step for metabolism of these disaccharides happens in your stomach, where you have to break the disaccharide into monosaccharides. So that hydrolysis is actually pretty slow. So you don't have a really, really fast supply of fructose like you do when you have fructose by itself. And that's the concern with high fructose corn syrup, is that it is fructose in its monomeric form. It immediately enters the blood, and there's this mass surge into the liver. The liver metabolizes it, and that ends up with fat synthesis. All right. Galactose. So galactose, we said, is present in milk because of lactose. So the idea here is we end up having a net energy yield the same as glucose, but it happens in a little bit different way. Okay, so galactose gets phosphorylated by this enzyme galactokinase, which makes galactose 1-phosphate. And here's the interesting part, is that galactose 1-phosphate is converted to glucose 1-phosphate, but not by an isomerase. Instead, it uses a transferase. So there's this enzyme galactose 1-phosphate uridyl transferase that uses this molecule UDP glucose as a substrate along with galactose 1-phosphate. And basically, it just swaps positions of glucose and galactose. So we start with galactose 1-phosphate, and we start with glucose bound to UDP, and we swap the sugars. We end up with glucose 1-phosphate and UDP galactose. The glucose 1-phosphate is readily isomerized to glucose 6-phosphate. This is an enzyme we'll see again when we talk about galactose metabolism. Glycogen metabolism. And then the UDP galactose can be easily epimerized to UDP glucose to make a net cycle that does not consume any ATP. Okay, so the ATP yield is identical to glucose. Again, just like with fructose, that makes sense. It's still a hexose. It still has the average oxidation state of carbon, the same as glucose. So it makes sense that we have the same energy yield. It just goes about it in a little bit different way. Any questions about these before we move on to TCA cycle? All right, cool. So I'm gonna